Open Science First Friday series, which is one of the initiatives of the Global Young Academy's Open Science Working Group. And the Global Young Academy is 200 young scientists who have an interest in using science towards the betterment of society. So there's members from all over the world, and Steph and I have been leading this group for the last year or so. And yeah, so every first Friday of every month, we have a workshop or webinar that is open to everyone. So please do keep posted on our next events. Our next First Friday, Kelly Woods and Katie Rogers will lead a discussion on multilingual publishing and metadata, like things like abstracts, at Open Research Europe and other open access platforms under F1000. This discussion will be summarized and provided as feedback to the European Commission to help guide future actions by these open access platforms. So if you're interested in hearing more about Open Research Europe or other F1000 open access platforms, or in just discussing multilingual publishing and how that impacts the open science more generally, please do join us. But today we are so grateful to have Tim Parker here with us to talk about a recent project he's been involved in. Tim Parker completed a PhD in biology at the University of New Mexico, a postdoc at Kansas State University, and a National Science Foundation research fellowship at the University of Oxford. He became faculty at Whitman College in 2006, where he has since held several leadership roles. Tim is an ecologist by training, and as an ecologist, he researches cultural evolution in songbirds, as well as demographic responses of plants to environmental change. But over the past decade, about half of his publications have focused on actually how we do science and on doing science better through making it more transparent, reliable, and reproducible. He's a founding member of SORT-T, the Society for Open, Reliable, and Transparent Research in Ecology and Evolutionary Biology. That's where I met him, and I know he's going to mention a little bit about SORT-T today. I'll also put a link in the chat. Some of you might be interested in joining. And he's also served on several other committees for that strive to make science better. He recently told me that this research he's presenting today is among the most important in his career. We had the opportunity to have some talks at a recent conference, and I wish it were a conference. And he was really excited about this work. So I'm really thankful that he's sharing it with us today. His team's findings are relevant beyond ecology and evolutionary biology, really for any field where there are people making decisions about how data are analyzed. In addition to asking Tim about his work, again, please do feel free to ask him about SORT-T and some of these other initiatives he's been involved in. And I'll help post some links to the preprint for this paper, SORT-T, and to our First Friday events in the chat. The preprint's already getting a lot of press, so yeah, you get to hear a little bit more about it today. So thank you so much, Tim, for joining us.
Yeah, thanks so much, Tasha, for that great introduction. Okay, so I am going to talk to you today about this recent project that we're just finishing up now. What's going on? I'm figure out why anyway, whatever. Same data, different analysts. It's meant what we call a many analyst study in ecology and evolutionary biology. And before I get started, I want to really emphasize the fact that this is a highly collaborative project. Not only are there a group of other folks who led this project with me. So in particular, I want to mention Elliot Gould, who's a graduate student, and Hannah Fraser, who are the two people who co-led this project with me. The three of us were the co-leaders of the project. I also want to mention Shinichi Nakagawa, who spent a lot of time contributing to the meta-analyses we'll see today, and Simon Griffith and Pete Vesk and Fiona Fiddler, who also helped with conceptualization of the project. But most important, I want to mention that this project depended on more than 300 additional people who contributed their efforts to either analyze data for the project or to serve as volunteer peer reviewers. And all of those people are all co-authors on the project. I don't remember now. The project has either 310 or 311 co-authors. Fortunately, the journal agreed to accept a spreadsheet of an author list and didn't require us to enter each author into the submission form, which is good. Okay, so what am I talking about today? Why can't I get this thing to get away? Hold on a second here. Okay, so I'm going to start off today. Well, I'm going to talk about divide my talk into four sections. I'm going to talk about heterogeneity in results among studies in ecology and evolutionary biology, and actually not just ecology and evolutionary biology, so some other disciplines as well. So just in general, the phenomenon of essentially differences in results. And I am going to talk about the study that I conducted, or that I conducted with those 300 other people. And then I'm going to talk about the implications of the study. So spoiler alert, we found diversity in analytical outcomes. So we'll talk about why is this a problem, and then we'll talk about what maybe we should do about it. All right, let's see, come on. Okay, so hold on one second. I'm trying to get this thing to... All right, whatever. Okay, trying to just influence what I'm looking at here on my screen. So heterogeneity in ecology and evolutionary biology. Ecologists and evolutionary biologists have known for a long time that results often differ from study to study. But this was quantified in a really important meta-analysis back in 2016, titled Heterogeneity in Ecological and Evolutionary Meta-Analyses. So that study looked at heterogeneity across a whole series of meta-analyses, and they found it to be quite high. So these measurements here, I-squared is a common index of heterogeneity often reported in meta-analyses, and it's the proportion of variance attributable to differences among studies rather than sampling error within studies. So we always expect there to be differences among studies, even if we were working in exactly the same conditions with exactly the same subjects, et cetera. If we went and redid a study and regathered the data, we'd expect there to be some variability in a system, in a probabilistic system. We'd expect there to be some variability just due to sampling error. So we're not surprised when we find somewhat different results among studies, but what this study showed us was that a huge proportion of the variability among studies is greater than we would expect just by sampling error. So something else is going on here. These levels of I-squared that this study found between 85 and 91, 92%. These are much higher than you would find in, say, clinical trials in medicine, for instance. So they're very high. There's a lot of variability from study to study in ecology. Let's see, once again. All right. So anyway, I already said that. Results vary substantially from study to study. So there are several potential explanations for this. True biological heterogeneity, methodological heterogeneity, and analytical heterogeneity. So true biological heterogeneity is the expectation probably of most ecologists. We just think, okay, the ecological world is really variable. We know there's a lot of contingencies. There's differences as we go out, into these, for instance, these different grasslands from around the world pictured here. We don't expect exactly the same processes to be happening in these different places. And so we don't expect exactly the same results in different studies. This isn't a surprise. This has typically been, the null expectation in studies. So when you read the discussion sections of papers, if they're putting their work into context and explaining differences between their study and some other studies, very often the explanation is some sort of biological difference. Oh, it was a wet year. It was a dry year. My system differs in this way. My system differs in this other way. So that's the typical explanation for any difference among studies, is a biological difference. But we know that studies don't use the same methods. So different researchers very often use different methods. And these differences could plausibly lead to different results. Ecologists have been thinking about this for a little while. For about 15 years now, there have been some important steps to try to address this. Maybe most notably is NutNet, or the Nutrient Network. This is a really cool project uh, known as a distributed experiment, where people from around the world have contributed to the same experiment, and to contribute to this experiment, they adopt standardized methods and and contribute their data to a central source. So this is something that ecologists are aware of, that there are other sources of heterogeneity besides biological heterogeneity. With NutNet, if there are different results amongst sites, it really should be mostly attributable to biological differences among sites. I say mostly attributable, both because, of course, there's still going to be sampling error, and also because even with the nutrient network where the methods are standardized, it doesn't mean that all methods are implemented exactly identically from study to study. But they're much more similar than in the average set of ecological studies. But what I'm going to talk to you about today is this third possibility, this third explanation for why results might vary among studies, and that's analytical heterogeneity. So with analytical heterogeneity, we know that different researchers often make different analytical choices, and these different choices might actually lead to different statistical outcomes. I think everyone would agree that this is a possibility. I think, however, people might not appreciate the extent to which it can be important. So Andrew Gelman, who's a well-known statistician, borrowed this term from literature, the garden of, or from the humanities, the garden of forking paths, to describe the almost infinite number of options that a researcher faces when analyzing a given data set. So when you start to imagine these choices, which data, which variables, how to transform the variables, which models, et cetera, it's very easy in a moderately large data set, say with 20 or 30 variables, it's very easy to rapidly approach a situation where there's literally, a million different choices for exactly how to run your analysis. So this is something that people in the world of metascience have been thinking about. So metascience is a discipline that I've been involved in now for about a decade, where scientists are studying their own work, this sort of the science of science. And some metascience is quite strong in certain social science disciplines where people are particularly concerned about the reliability of their discipline. So, for instance, social psychology has produced a lot of metascientists because of, I think, real valid concerns about the quality of some research in social psychology. Anyway, meta scientists in the social sciences started a few years ago doing these many analysts project, where they asked a lot of people to analyze the same data, to answer the same question. So they provide a data set, they provide a question, and then they invite anybody who wants to, or they invite a certain subset of people to analyze the data to answer the question. So here are three figures from three recent many analysts study. That 2018 Silberzahn et al. Paper was the first one, and then these other two are two other recent ones. When we first started planning our project, the only one that had been published and that we knew about was the Silberzahn et al. Paper, and then while we were putting together our proposal for this project, the Bresnow et al. Paper, the Bresnow et al. Paper was released as a preprint. What you can see with all three of these graphs that I've shown you is that results have varied among these studies. I'm not going to get into what the research questions were in each of these three studies, but you can see in each one, these are forest plots, which are typical plots presented in meta-analyses. And you can see real dramatic variability in the results that different teams of analysts produced. In these graphs, each point is a different effect produced by a different group of analysts, or a different single analyst. So quite a bit of variability from analyst to analyst here. Okay, so um, so we know from the social sciences that analytical flexibility can drive heterogeneity. What about in ecology and evolutionary biology? And this is where we're going to get into thinking about this study that we're just finishing up with. So we actually chose to do our study with two data sets and two questions. So our first question was, to what extent is the growth of nestling blue tits influenced by competition with siblings? And for this data set, to answer this question, 79 teams submitted analyses with a total of 135 usable effect sizes addressing the target question. And this, I'm not going to get into the details of this data set. I'm happy to answer particular questions about them after the talk if you want to. But basically, we had 30 some odd variables, and they came from 452 nests of these birds, these common European songbirds, blue tits, and 332 of those nests were part of a brood size manipulation experiment. And the data came from my postdoc in Oxford. Okay, the second data set, or the second question was, how does grass cover influence eucalyptus species seedling recruitment? So maybe most of you know, eucalyptus is a common genus of trees in Australia. And so these data come from Australia, from a restoration ecology study in Australia. And 68 teams submitted analyses with a total of 81 usable effect sizes addressing this target question. And basically, these data come from places in Victoria, in southern Australia, where folks were looking at factors that might be influencing the success of eucalyptus seedlings growing in places where restoration ecologists were hoping to restore eucalyptus woodlands. So again, about 30 variables from, in this case, there are 351 of these 15 by 15 meter quadrats spread at 18 sites around regenerating farmland in Victoria. Okay, so what did we find? Let's just jump right into it. So here's a forest plot, and I'm going to show you most of our results in forest plots. These are the blue tit data. So let's look at this graph for a minute in case you're not familiar with forest plots. We've got our standardized effect size on the x-axis. So the standardized effect size is this ZR or ZR Fisher's Z transformation of R. It's really common in meta-analysis. And it's very similar. So R being the correlation coefficient, which I think most people are familiar with. The values of ZR are very similar to the values of R. They're just sort of stretched out at the ends. So R is obviously bounded by 1 and -1, and ZR is not for improved performance in statistical analyses. So at ZR of 0.5, negative 0.5 or 0.5, it's about equal to R. But by the time you get out here to ZR of 1, you're at about R of 0.8, or in this case, -1, -0.8. So what do we see here? So on our x-axis, we see this value of 0. This would mean that there was a point that falls right on this line. Where'd my cursor go? A point that falls right on this line would be a point that found no relationship between a measure of sibling competition and a measure of nestling growth. So a point that falls below the line would mean that greater sibling competition led to reduced nestling growth, and a point above the one line, a point in the positive realm over here, one of these points found a positive relationship such that with greater nestling competition, nestling growth increased. Our meta-analytic average is right here. It's about 0.35. And each one of these points represents a separate effect size submitted by one of our analysts. And you can see that it's quite variable. It stretches from around positive 0.4 down to greater than negative 1.5. But most effects range from about positive 0.2 to negative almost negative 1. So almost continuous variation across that range. And we had a really high I squared of 97%. As a little aside, I will say the interpretation of I squared is a little bit funky here because all these data points are based on more or less the same data. So really, the interpretation here would be that if these points had actually come from different data sets, then we could actually interpret I squared exactly the same way we do in a meta-analysis. But it's a good sort of index just to show just how incredibly variable these results were across a wide range. All right, so let's see here. So here are the results for the eucalyptus. The x-axis is the same. The pattern of variability you'll see is different. First of all, the meta-analytic average is much closer to 0. And in fact, if you look here at this point down here at the bottom is the average and the 95% confidence interval, which you can see overlap 0. So we're not particularly confident that this mean is anything other than 0. And the overall most points are the range of variability across most points is actually lower than the range of variability across most points in the blue tip data set, but the overall range of variability is actually greater. This is just showing there's like an outlier. There's an outlier here of about negative 0.47. So the overall range of variability is higher, and there's a few pretty substantial outliers, but the bulk of the points the variability is narrower. So let's see. So the I squared here is 98%. Okay, so now that we've seen what I think are actually sort of the big results from the study. I mean, this is what most people are interested in when they're seeing these results. I do want to take a deeper dive and think a little bit more about these results. So first, I want to ask this question: why do we see such substantial heterogeneity? And then also think about why does this heterogeneity we are observing actually differ between these two data sets? Because I think it really does differ between these data sets. So I'm not going to give a comprehensive discussion of either of these questions here today. I'm going to talk about a couple of different ideas, or several different ideas. The first one, but anyway, yeah, not try to be comprehensive. So why do we see such substantial heterogeneity? So it could be that there's variation in the quality of analyses, and it's interesting, among the people who contributed analyses, when it came time to write up the manuscript, there were some of the 300 contributors were absolutely convinced that this was all about just quality of analyses, and we're actually saying, like, why aren't you devoting more space to talking about the quality of analyses? This is clearly just that there are people who know what they're doing and there's people who don't know what they're doing. So to some extent, that probably is true. Like, some of these analyses are poor quality analyses. So for example, the two extreme outliers on either ends here in the blue tip data set come from the same team, and their results of collinearity in an analysis. The person who provided these effect sizes really had some misunderstanding about drawing inferences from effects in analyses when they're highly correlated covariates. So those outliers, absolutely, in this case, due to poor quality analyses. But so at least some of this variability is due to poor quality, but I think most of it is actually not. So here's for the eucalyptus data. Remember, I mentioned that we had volunteer peer reviewers. So about half of those 300 volunteer contributors to this project were peer reviewing methods. So Each method section was peer reviewed by about 4 volunteer peer reviewers. And by the way, they weren't peer reviewing the results, just looking at the methods. And one of the things those peer reviewers did is they rated each analysis as publishable as is, publishable with minor revisions, publishable with major revisions, or deeply flawed and unpublishable. And so, first of all, you can just see that there's a huge overlap in these distributions in terms of the standardized correlation coefficient, the ZR. So that's on the x-axis here. That's just the same x-axis as those as our funnel plot, as our forest plots that we looked at before. So we also did some other things. So remember that the I-squared here is 98%. We tried, well, what happens if we drop the deeply flawed and unpublishable studies? What happens then? Well, our I-squared does go down somewhat, but it remains pretty high. If we go ahead then and drop publishable with major revisions, it doesn't really change very much. It stays about 75%. So that suggests that maybe analytical quality isn't very important here. There's an important caveat, which is that inter-rater reliability among these peer reviewers was near zero. In other words, what the decisions of one reviewer didn't predict the decisions of other reviewers really at all, which is kind of striking. And we're actually in our ample free time about to work on a paper about that as well. But I want to provide one other piece of information that suggests to me that analytical quality is probably not the main explanation of what's happening here. So we also asked analysts to rate their own statistical expertise, and the 2 highest ratings they could provide were expert, in which they said that they could teach an upper level statistical course and could explain the theory and the mathematical details behind each analysis, or highly proficient, which was a step below that, in which they could readily explain assumptions. And let's see, I don't remember the full set of requirements for highly proficient, but they were the 2 highest levels of analyst self-rating. And so we kept only analyses produced by teams that included at least one person who rated their statistical expertise as highly proficient or expert. And we found, this is the results from the BlueTit data set. We find a distribution that looks almost exactly the same as the distribution from the full set of analyses. So again, it really doesn't look like analyst expertise is a substantial component explaining the variability, or I should say lack of expertise. I do also want to very, very openly acknowledge that I don't think this is the last word on the subject. I think there are opportunities that we're pursuing actually to try to answer this question more thoroughly, but it's probably going to have to be with another data set. Okay. So some other possible explanations for why we see substantial heterogeneity in this data set. One is that there are a small number of variables with large and divergent relationships. In other words, it really just depends on which variables you include in the analysis. And if you include certain variables, you get certain effects. And, let's say large positive, and certain other variables, you get other effects, let's say large and negative in the opposite direction. So we addressed this with what's called a specification curve. So here we show that same forest plot that I showed you before, but now it's turned on its side. So you can see what was the x-axis is now the y-axis, and we can see this distribution of effects from different analyses. And then down below, we can see the different primary independent variables. So the particular predictor of meant to be the index of sibling competition there, the independent variable, and then the dependent variable below that, which is the measure of chick growth. And we can see, well, what do we see? We can see that there are at least some variables, like this particular contrast from experimentally reduced broods versus unmanipulated broods. There are particular variables where the results skew one particular way. So here we see that, all these really small effect sizes, or not all of them, but many of the really small effect sizes and the effect sizes where there is actually a positive relationship between sibling competition and growth. Some of these come from this particular independent variable, and basically all the cases of this independent variable are associated with pretty small or positive effect sizes. Maybe we see the sort of the opposite here, although there's more spread in these points. With when we look at another way to measure this, which was net brood size manipulation. But I think we also so to some extent there are a few variables that point in one direction or another, but you can also see that a lot of these variables are found across the spectrum of effect sizes. So most variables, you can find them in models with everything from really extreme effects in one direction to really extreme effects in the other direction. So another issue is the analysis of subsets of the data. So obviously, if we subset the data, we can potentially get different different patterns with different subsets. And to some extent, this seems to be true. We have some evidence of this. So here, I don't know if you've just seen this here, but our sample sizes have popped up here at the top for these three most extreme outliers in the eucalyptus data set. I'll say that the maximum sample size here from this data set was 351, I believe, 351 or 352. Most of these studies had sample sizes above about 340. These extreme outliers are all from subset. Actually, that 90 is a subset, the 117 is a subset, the 18 isn't actually a subset. They've averaged. There are 18 different groups of sites, and they averaged within each of those 18 sites. So to some extent, subsetting seems to be driving things. And this demonstrates that the effects of subsetting can be quite large. Like if you choose a substantially smaller subset of the data, you can find a very different pattern. So here are looks at some more evidence associated with subsetting. So notice here, this is the eucalyptus data set. These are sample sizes from individual studies. Notice there are some blanks because some people were not entirely transparent with us, and we don't know exactly what sample sizes they had. But notice that most of these bars go up to about 350. Like most of these bars are working. Most of these samples are based on pretty much exactly the same sample size. But when we go over here to the blue tit data, we see much more variability in sample size. We see some trends. Like we can see there's about there's some trends here. These are folks who used just experimentally manipulated broods. But we can see more variability, and that's consistent with the greater variability we see in the effect sizes in the blue tit. So maybe subsetting of data has a broader explanatory power here. Another possibility is inclusion or exclusion of covariates, especially those with large effects. Other possibilities include model structure, model type, etc. And there certainly are other possible explanations as well for the variability, for the heterogeneity we've observed. I want to take, so including actually a very important one, which is vague questions that allow for diverse model specifications. So actually, I mean, this is certainly, depending on how you look at it, this is a major explanatory factor for our results. So this is a paper that was published in response to the very first many-analyst study in response to that Silberzahn et al. Paper from 2018, where they said, among the main points of this Osberg and Brutal paper was that the researchers had asked too vague a question, and it gave researchers too much flexibility in which model specifications they could choose. And that really what researchers just, that was the problem. Like it was just, they asked too vague a question. Um, I think that this is, there's important truth to this, but it's also an interesting, it's an interesting criticism because the question that was asked, for instance, by those researchers in the Silberstein et al. Paper, and the questions that we asked in our research are meant to reflect the sorts of questions that researchers in this field ask. So certainly in ecology and evolutionary biology, people ask a lot of vague questions. So here's some questions asked by 5 recent meta-analyses in ecology and evolutionary biology, and you don't have, I don't know, pick one to read. But the point is that none of these map really precisely onto an exact model specification. Any one of them could be specified by many different models, specified in many different ways. And this is simply, it's the level of prediction that theory typically makes in our discipline, and it's the way that we think about biological questions. And so I think it's important to keep in mind that the level at which we ask questions predisposes us to generate variable results. But I think it's also I think it's just wrong to think that we can simply just get out of this problem just by asking really precise questions. For part of the reason is, even if individual studies asked really precise questions that could only be specified by a very small set of models, the people who read those papers and who are trying to draw general conclusions about the world are going to lump those very specific papers with other papers in meta-analysis, for instance, and in reviews, and think about them in more general terms, and are still going to say, oh, well, these studies found different results. Okay, so diversity and analytical outcomes. Why is this a problem? So maybe it's obviously a problem to you, or maybe not. Let's think about why it might be. So when individual results replicate poorly, in other words, when we don't get the same result from study to study, and that's caused by, for instance, analytical heterogeneity, it means that when we see a result in the literature, we can't rely on that individual result. We can't assume that that result is actually something that we would find again if we looked again. So I think that is a reason for concern. It's certainly a reason to be more cautious. I think I will say that so some researchers that I talk to say, oh, well, we knew that already. Like we didn't, nobody, well, I don't know if they say nobody, but they say we knew that already. It's not a surprise. Of course, you can't rely on individual results. And fair enough. I mean, if that's how you operate, that's great, because I think that you didn't need our study to tell you that you shouldn't rely on individual results. However, my sense is that really that's not how most people operate in our discipline. We may, if pushed into a corner, we might admit that, yeah, you can't rely on individual study. But if you look at how people talk about individual studies and things they say and write about them, it certainly looks like we often draw pretty strong conclusions from individual studies. So I think another reason why analytical diversity and analytical outcomes is a problem is that it provides substantial opportunity for bias to skew reported results. So, and this is potentially even a bigger reason for concern. So we know that there is bias. So I'm just going to show you results from a few studies, some of which I was involved in. So I was involved in a study of about 5,000 authors. Well, yeah, survey. We asked about 5,000 authors and got about 800 or between 800 and 900 responses about their research practices in ecology. And for instance, so this graph here, if you can, I don't know, turn your head to the side, you can read that it's the x-axis or the y-axis have failed to report a non-significant result at least once. So this is somebody they found some result that was not statistically significant, and they just didn't tell us about it in the paper. And you can see there, so that's ecologists. Those two bars are one bar is ecologist, one bar is evolutionary biologist. And a large portion of ecologists and evolutionary biologists admit to having done this. Here's a similar sort of thing, but this is either non-significant covariates not reported in the left two bars, or some models not reported in the right two bars. Again, lots of people admit to having done this. So it certainly looks like, so that's evidence that people, in fact, are doing different analyses, therefore generating different results, and then reporting a certain subset of those results that tell the best story or most convincing or something. And we actually have growing evidence in ecology and evolutionary biology that this actually leads to bias. So there's several papers published just like within the last year or so. So this paper, publication bias impacts on the effect size, statistical power, and magnitude and sign errors in ecology and evolutionary biology by Yang et al. I am an author on that paper. I contributed some important stuff, but not. Certainly, the bulk of that work was done by Yafeng Yang. Another paper, empirical evidence of widespread exaggerated bias and selective reporting in ecology from Kimmel et al. In Nature, Ecology, and Evolution. And then a preprint, which has yet to be, well, it's a preprint yet to be peer reviewed, or at least yet to be approved by peer review. More than half of statistically significant research findings in the environmental sciences are actually not. So, and then I co-authored a review a little brief review and a news and views piece of these papers recently, Exaggerated Effects in Ecology. We have pretty darn good evidence that that effect sizes in ecology are exaggerated. In other words, the raw material for bias that we're seeing with our many analyst study, and the fact that you can analyze data different ways and get different results, that raw material is actually ultimately producing bias. And depending on the study, let's see, do I have a number here for that? No, I don't. But depending on the study, the average effect size, or depending on the discipline and the type of statistical analyses, average effect sizes can be as much as probably double the true effect size in the ecological literature. All right, so it's a problem. I think this analytical heterogeneity is a problem in the current environment. What should we do about it? Again, I don't want to claim that I'm making a comprehensive set of recommendations here, but I'm just going to review a few quick ideas. Economists like to talk about robustness checks. Ecologists sometimes talk about sensitivity analyses. These are situations where we test several alternative models to try to see if we get sort of the same result. In practice, they're generally presented to confirm the reliability of the given analysis. So there's room for bias in terms of which robustness checks you present. Some limitations here: a small number of models is going to provide limited insights into the scope of possible outcomes. There's, as I just alluded to, there's still a risk of bias unless you've somehow constrained beforehand through something like pre-registration or registered reports. You've somehow constrained the set of robustness checks you're going to pursue. Another more sort of thorough response would be a multiverse analysis, so where you can specify a broad universe of plausible models. And these are often combined with a specification curve where you map particular specifications onto a range of effects. So I already showed you this specification curve from our many analysts work. It's important to note that, of course, this range of effect sizes here doesn't come from a true multiverse analysis, because a multiverse analysis would be an analysis where we systematically identified possible specifications and then implemented them ourselves. Here, of course, it was different analysts, and the fact that they varied just happened to be, just happened to occur by chance. But this specification curve, this is a specification curve where we've got this range of different specification information, and we could have a lot more detail here than just the independent and primary independent and the dependent variable, where we map these different aspects of model specification onto effect size. So some potential limitations of multiverse analyses with specification curves is that determining the scope of theoretically justified model specifications is subjective. There isn't one sort of set. We can't all agree on what range of specification space we should examine. So this isn't a total solution to this problem that different people specify models differently, because we're going to still specify our multiverse analyses differently. Also, the more comprehensive you seek to get in terms of specifying different model possibilities, the harder it is to make sure that you're actually retaining some sort of statistical or theoretical validity, because there may be lots of model specifications out there that make sense, but other ones that actually don't make sense, that no one would actually code because they're inherently contradictory because of biological reality or something. And another practical issue is just that you can run up against potentially coding literally hundreds of thousands or millions of different alternatives, which is overwhelming computationally, and also just how do you report that? How do you make sense of it? I will point out that Simonson et al. Recommended essentially a random draw from the set. If you have a huge set of possible specifications, you don't actually do them all. You take a random draw from them and then sort of sample across the range. In ecology and evolutionary biology, for more than 20 years now, certain subdisciplines have really embraced model averaging. So this is a possibility. There are potential limitations of model averaging. Burnham and Anderson recommended just looking at 20 or fewer models with strong a priori justification. So they're advocating exploring a relatively small array of specification space. And then there are more recently, some authors have pointed out that there are some real problems averaging regression coefficients across different models. There are a number of different situations that are quite common in ecology which invalidate this type of model averaging. And then again, whenever we're choosing multiple models, there's subjectivity involved. Anyway, this is just a rehash of just the fact that with both of these, there's many, many different options. And when you have, when you're faced with a situation where you're doing many, many different types of analyses, this really raises the stakes of how difficult it is to conduct a statistical analysis. So we could ask more precise questions. I already kind of addressed this. I think that there's, when we ask super precise questions, there's a mismatch between the formulated question and the actual question we have. And then another possibility is caution and humility in interpretation of results. So we could just stop writing papers that suggest we have the conclusion. The problem with this is that there's incentives don't really favor humility in paper writing. Maybe we could redesign incentives so that they do, but they don't really right now. And of course, we'd still be left with uncertainty. So I just want to emphasize that I think the way forward is unclear. There are a lot of different options for how to respond to this knowledge that analytical heterogeneity is major and important. I think multiverse analyses are wonderful potentially if they can be managed, but they have some real difficulties. I think pre-registered robustness checks are probably going to be more realistic for a lot of people. And that this is not I just again want to emphasize there probably are other solutions. There may be other better solutions than the ones I've talked about here today. Okay, so I think in ecology and evolutionary biology and in any other discipline that has some statistical similarities with ecology, there need to be discussions and debates, and people need to work together to figure out what the way forward is. All right, before I take questions, I just want to mention Sortie. Tasha mentioned Sortie at the beginning. Sortie is the Society for Open, Reliable, and Transparent Ecology and Evolutionary Biology. We're an international organization that we bring together researchers who are committed to improving the quality of research in ecology, evolutionary biology, other disciplines. Sortie, we have an annual conference. We have We have a preprint server, EcoEvo Archive. Our preprint server is not. I will point out bioRxiv also exists. We don't control bioRxiv. That's separate. Our preprint server has some options that bioRxiv does not. And so I think, depending on what you want to do, our EcoEvo Archive may be better for you. Anyway, we're a community of people who are interested in improving ecology and evolutionary biology. And I think Tasha put a link in the chat. But anyway, we'd love to have you if you want to join us. You come check out our website at sortie.org. And then, I'm happy to take any questions about this work. And I'm going to stop sharing my screen, I think.
Yeah, and really quick with Sortie, the membership fees are very, very reasonable. So yes, please, a round of applause. That was really so interesting. And I have several questions, but I want to let others ask questions first. So did anyone have any questions for Tim about the study or its implications or how it might relate to your field if you're not an ecologist? Yeah, go ahead, Christoph, please.
Yeah, hey Tim, thank you for this great presentation. Very interesting, and I think the stakes are quite high. Um, so my question would be, like in the context of the blue tit, like data, I could imagine that basically the effect size switches like science when you look at small broods versus large broods. So like for example, one additional like individual in like small broods could be actually advantageous, whereas in a large brood, one additional like individual is like fatal or like very deleterious basically. And then how do you extract an effect size from such like a model that accounts for that?
I'm not sure I understand your question. So you mean like, is this a, could you rephrase it? Because I'm not sure if you're asking about, you said, how do I extract an effect size?
Yeah, so
Is that
basically,
a statistical question or is this a theoretical question? What's the,
well, this yes, basically my question is like if the effect like depends on like another variable, like for example brood size, right? Then you would have like a negative effect for small broods and a positive effect for large broods basically. And like the model that you fit then basically like has this additional like variable that influences like the entire model. And I don't see how you can like like how you can extract one effect from such a model because it's context dependent in the end.
right. Well, so so you can, obviously there's, at a practical level, you can, extract a partial correlation coefficient or whatever, the partial correlation and call that your effect. Whether you want to consider, there's the sort of the extent to which that's theoretically justified or not. And obviously context dependence is huge. I think, if you, I think using those different effects in our meta-analysis is justified because I think it's what people often do in the literature. Like you go in and people will, they'll have a complex model and they'll say, oh, here's this effect. That is drawn from this more complicated model. In the case that I was pointing out, it was from an experimental manipulation where it was, if you removed chicks from a nest and compared that to an unmanipulated nest, what was the difference? And the difference is basically averages about zero. Like the difference is, depending on how exactly you specify the model, it's about zero because it's actually not in that case the biology. It's not really helping the parents any to reduce the brood size. And then if you however give them additional chicks to feed, it's then it does start to limit their ability to feed them. But anyway, that was an aside. I don't know if that answered your question. I hope so, but
Yeah, like I'm not quite sure if I understand, but I think I will leave the stage for other people to ask their questions. Thank you.
Thank you so much, Christoph. Other questions? Steph, go for it. Oh, you're muted.
Yeah. So I'm wondering, you started the talk by saying that we were basically as ecologists doing way worse than clinical research and such in terms of the generalizability of our work or the, yeah. my question is, what other lessons could we learn from clinical research that we might be able to apply in ecological contexts?
Yeah, I guess I wouldn't characterize it exactly the way you characterized it. Like I wouldn't say I don't think we're doing worse at generating generalizability. I think we just I think the difference is that the clinical research is working with much more under much more sort of constrained systems. I mean, first of all, the studies they're combining are going to be all on people, for instance. So we're all working in a single system, and they're working, they're trying to they're generally, often working in relatively highly controlled experiments. They might be looking at the effect of like a particular drug, that they give it a particular dose. And so there's just a lot less variability, I think, often inherent in the system. So I don't know. I mean, there is certainly heterogeneity heterogeneity that researchers are really worried about in clinical research. It's just not as high as it is in ecology and evolutionary biology. And I'm not sure that there's a lesson there other than we just work in these fundamentally varied systems. I mean, arguably a lesson could be, and I mean, this is something I've sort of thought about on and off for a while. I mean, I think maybe a lesson is we work in it's harder to draw general inferences in ecology and evolutionary biology than we often think it is. So a lesson is like we should recognize that this is more difficult. And maybe a lesson is we should try to do more stuff like the stuff that happens like with NutNet or whatever. We should try to be, instead of everybody going off and working in their own system, maybe we should have more networks where we're trying to work together in the same or similar systems and work more similarly. I think that sort of is frustrating to people who really enjoy going off and working in their own pet system. And I'm not saying that that's really a viable suggestion, but I think, if you're looking for a lesson from that clinical work, then maybe that's the lesson.
Yeah, thanks.
Any other questions? Svenja, yeah.
Hi. So I was actually one of the people who, or one of the team members who submitted one of the data sets, the BlueTit data set.
Yay,
so I was really excited also just to hear kind of how you're communicating this study to a wider audience. And something that I know from myself and many others, so I'm a fairly young lecturer to start lecturing, is how do we train people up to have the statistical knowledge to, let's say, do more robust analysis, to when you choose your model, you choose the model that is, because I feel like a lot of it is also just having the statistical knowledge and the confidence in selecting the right model rather than, let's say, using the approaches you've maybe been trained up to use and, a particular set of analyses. So yeah, I feel like there's the kind of whole training question is something that I'm thinking about a lot because I would like to learn more, but it's also really hard.
Yeah, I mean, I think it's a really good question, and I don't think I have a really satisfying answer for you. I do think that I mean, even though I sort of addressed that question of, how much of the variability is due to poor statistical knowledge, and I said, well, maybe not really very much. I mean, I think that there is some of the variability is due to that. And clearly better training would address that. And I'm not sure what the answer is. And I think it also varies, like just the academic systems vary so much from country to country, and I that there's, I think some systems might be better at doing this than others. I not that I think that I know which ones are better, but I mean, I think, that there's probably, maybe if we looked around and we tried to assess in which systems people seem to have ended up with the best statistical knowledge, maybe we could figure that out. I don't know. That's my empirical answer to you. Like, well, let's figure out who's got the better answers and what are they doing right? Because I don't know.
I will say I appreciated the comments about humility, but I am a person who is never certain in myself and does a lot of may in my writing and stuff, and reviewers do not like that. So whenever I've tried being humble, subconsciously, even because of uncertainty myself, I've gotten a lot of pushback from reviewers. So it's one thing to like teach students or talk with students about this humility piece, but then, like you said, it's not often, something that you get, you know, published for
Oh
or
yeah.
whatever. Like there's pushback.
Yeah, no, and I mean, there's incentives. I mean, one thing I didn't really talk about, it just sort of alluded to it briefly. I mean, but all this work is done in cultural context and in the context of the incentives imposed by the institutions that sort of govern how science gets done, and that includes things like the universities or research institutes where we work, the journals that publish our research, the funders that fund the research, and all that stuff is really, really important. And right now, incentives often work against reliability in various ways, and they certainly work. They often work against that sort of against humility. I mean, editors, it's not hard to find stories of journal editors who tell people to remove certain analyses, remove this analysis. It doesn't help the story or whatever. Like, I mean, I've certainly had that happen to me, and I know lots of other people have had that happen. Sound more, be more, make your abstract draw a stronger conclusion or whatever, all those sorts of things. Yeah, that doesn't help. But
yeah,
yeah,
I know we're past time, but I still hoping to ask you a question. I don't know if anyone else though before had any questions. I was curious
So
about
if I may just
whether,
quickly, sorry,
go
just
ahead.
one thought I find really fascinating. I think this whole idea of telling a story, I mean, I always hear, what's your narrative? What's your narrative? And I mean, what you said, I've done the same, right? I haven't included some results because it wouldn't fit a narrative. And if I don't have a narrative, I can't pitch it to an editor. So I think the kind of whole thing of like science communication, which I feel like where this is coming from a lot, is super important, but maybe there isn't always a narrative, and maybe we just have to accept that, look, this is really messy, but we need more, like, as you say, variation, messiness, whatever you want to call it. And sorry, I just want to comment on this quickly. This kind of storytelling, I understand as a reader, but sometimes I find it also really frustrating as a researcher because there is just no clear story often.
Yeah, absolutely. It's a messy world. And yeah, I think the desire for clean stories is it absolutely influences the distribution of effect sizes in the literature, and then makes us think we know things that we don't know.
Stephen, are you okay? Or Tim, are you okay to stay on for a couple more
Yeah,
minutes
that's
to answer
fine.
questions? Okay, cool. Stephen, please. Thank you so much, Pena.
So, hi, thanks for the brightening talk. I just wanted to ask, do you see a future for computer-generated analysis? I mean, all we researchers make different decisions. Will we get to a point where we mainly put our research questions into a computer and let them design and come up with all the different models, especially in your, what was it, multi-model verse context? I mean, computers could help to draft out not correct, but all the different analysis for us or something. So there's something like this.
Yeah, I mean, it's an interesting question. I think right now we're not there. I mean, what I'd be concerned about right now is, right now, I mean, I haven't tried this, but I mean, just from what I know about how, generative AI works, if we asked AI to generate analyses, it's just going to look around and see what other people have done, right? It's just going to be like, oh, well, somebody else did it this way. So I don't know if I don't know if, and people have obviously done these a lot of different ways. I mean, so like maybe we could generate, maybe we could, maybe it's one way to generate multiverse analyses. We could, instead of us trying to code them, trying to think carefully about them, we could just ask AI to generate a bunch of them. Obviously, that's going to be heavily rooted in just what other people have done. And so, if that's good, then, if that's a sample of what's plausible and reasonable, then sure, then we could do that. I think it just depends on how much bias there is on what's already been done. And maybe there's not a lot of bias there. I don't know, maybe because the bias is more sort of an outcome. And if you're separating the modeling choices from the particular data set, then maybe that's okay. But I will say, I mean, I think one of the challenges in general about one of the biggest challenge, and I didn't really emphasize this, I mentioned it briefly. One of the biggest challenges with multiverse analyses is really there are, millions of ways to specify a model, but it's very easy to just say like, well, what's all possible combinations of all whatever, and you just get a million options or whatever. But of course, many of those aren't particularly biologically plausible, and so and so I'm not sure. I don't know to what extent working with AI would help us navigate that. My sense is maybe, I mean, maybe it would. I don't know. I haven't worked very much with generative AI. I don't know. I'm too old, I think. I don't know. Like I mean, I think there's a lot of interesting things that could happen there, but it's not obvious to me that it's a solution right now. But whatever. There's lots of stuff that isn't obvious to me. So maybe it is, and I just don't see it yet.
Others? So one thing I was thinking about a lot, so I've worked in a couple of systems now where I've worked with people who have worked in those systems for decades, and they have a lot of kind of anecdotal, I know that this is this way. So I worked in a penguin lab for a while where the PI had very, preconceived notions about what the answers would be to certain analyses. So like whenever I would do something, she'd be like, "Oh, I knew that," or like, or would want you kind of almost to prove with models what she already knew, which she. And then I'm not trying to undersell, like having a long-term anecdotal knowledge is really important deciding like what's biologically realistic, right? But I was curious whether you asked people. So I had an initial gut reaction to the blue tit response because I've worked with birds, right? The eucalyptus data, I was like, I don't know what I would expect to be the case in either direction. But like the blue tit data I came in when you were showing that with expectations that increasing brood size would have a negative effect on variables like mass and wing chord and stuff like that. So I was curious if you asked whether like people had at what people's experience was in that system or in similar systems, and whether that biased kind of what their outcomes were, like if people who had worked on bird stuff for a long time were more likely to see a strong negative effect. And then also thinking about the reviewers too, like if reviewers have more experience kind of in that system, were you more likely to see that like a reviewer who had worked in a system for a long time had more pushback to there being a positive effect of changing brood size? Like what kind of this longer term experience on these types of systems and preconceived notions, I just wonder how much that affects the outcomes, if that makes sense.
Yeah. So, I mean, those are good questions. I mean, so I will say the reviewers didn't actually see the results, so they just saw the
Oh, right,
method,
right,
so they
right.
wouldn't have been able to push
Right.
back there. But the short answer is that we didn't ask people about their expertise in the system. We just
Mm-hmm.
asked them about their statistical expertise. And the longer answer is, yeah, I think absolutely. So expertise in the system could influence outcomes. I have two things to say about that. I mean, one is so in some of those social science, in at least one of those social science meta or many analyst studies. Well, there's one that's actually not really social science. There's one that I didn't show you results from is from, well, it's kind of at the edge of, I don't know what it is. It's like social neuroscience or something, but it's people using fMRI, so this brain
Mm-hmm.
imaging stuff to answer certain questions. And everybody who contributed to that was basically some kind of fMRI expert. Like there was a pretty high level of expertise in the people
Mm-hmm.
doing the different analyses, and they still found
Mm-hmm.
huge heterogeneity. So I'm not sure that expertise in the system alone is enough to prevent it. I do, however, think that it's an important question to ask. I mean, I have a grant proposal that's in review right now, where one of the components of the one of the components that we're looking at is how experimentally is how does expertise in this system or very similar systems influence the degree to which heterogeneity emerges. My suspicion is it probably influences a little bit, but that it won't influence it probably as much as people would like to think that it would. I don't know. That's just my guess, but just based on stuff that I've seen from the social science literature.
Thank you. Alrighty, thank you so much, everyone. And again, please do check out the preprint and sortie, and join us again in about a month to talk about the future of multilingual publishing on open access platforms. One more round of applause for Tim. Thank you so much for staying after.
Thank you. Thanks, everybody. It was fun.